I was saying that the community is really nice. I was driving through the long road, and there's so many trails and paths to walk through. It's really nice for like a Gainesville in the actual city. Nice. Hello. Okay, like higher pitch or lower pitch is better. Is this okay? Like in a voice? Okay. Oh, yeah, perfect. Yeah, that's great. Thank you.
Yeah.
Oh yeah, I'll pass it around. I understand. I understand.
Yeah.
Come
Hello,
on in.
Mary. How
Yes,
are
we'll go
you?
ahead and get started.
Good morning.
If you have a cell phone on you in the room, if you could please silence it, would be greatly appreciated. If you are on Zoom, you are muted. Please stay muted. If you have questions for the speaker, please put it in the chat and we will respond to that after the presentation. Walter Wynn is going to introduce our speaker for today.
Good morning. Welcome here in the audience and on Zoom. Our speaker today is Eric Trotman. He comes from Barbados. If you don't know where that is, it's due east of St. Vincent and the Grenadines. If you don't know where that is, Google. So, he's a lucky guy because people usually have to do their research in Africa or Canada or somewhere. He has worked it around so that he does his research in his home island of Barbados. And today's going to tell us about why the great snails are a problem. So, Eric, welcome. Thank you.
Thank you. I appreciate it, Walter. I have a couple things here. I'm wondering if I should wait till the end so people see them. Yeah, okay, I'll do that. Okay, good morning. Thank you for having me. Can everybody hear me? Is this a good speaking space for everyone? Yes. Okay, great. So today I am going to talk about the globalization of the giant African land snail. It's going to be a synopsis of my entire dissertation work. Slower. Of my entire dissertation. So I am going to tell you a little bit about what my dissertation focuses on and the focus of the research. So I am focusing on science guided by this One Health framework. This One Health framework connects people, animals, and the environment. It's a way to include all voices of a problem, basically. Because in conservation, in wildlife issues, there are not a lot of um, areas that focus on every individual sector. Sometimes it'll only focus on the animal, or it'll only focus on the people, or it'll only focus on the landscape. But it's really important to focus on all three of those things to combine and to solve a problem at the end of the day. So my PhD, I am in the 5th year. of my PhD right now. The first and second year, I was studying a aquatic snail in Florida called Planarabella trivolvus and a parasite. Um, I'm talking about a lot of parasites in my research, so bear with me. That can cause swimmer's itch in snails. And then in my third and fourth year is when I then pivoted to research in Barbados to focus on the giant African land snail, or Lysacchatina filica. And I'll tell you a lot about it as we go along, so bear with me. And I have some live samples of the snails as well. And then, as Mary would know, in ceramics, I made a replica of one of their shells that I'll show as well. So, yeah. Okay, so Lysacchatina filica, or the giant African land snail, is the top 100 most invasive species globally. So it's listed in the top 100 most invasive species. So of animals that have moved across from their native range into an inhabited range somewhere else in the world where they're not natively found. So they exist in places where there's high humidity. So that's about 80 to 90% of rain throughout the year, very much like Florida. However, in Florida, where we are right now, we are too far north for this snail to exist because of the freezes that we do get in December's in the winter season. But right below our range, the giant African land snail can exist in. And we have actually had introductions of the giant African land snail in Florida, and they have spent about $10 million in 1969 to eradicate this snail. However, it came back in 2011, and they're still spending money to try and eradicate it. It is only in small pockets across in these areas, so in Pasco County, in Broward County, and a couple other counties. And when these snails have an outbreak, the Department of Florida Department of Agricultural Services is really good about eradicating that population, and they quarantine the area that they're in as well. So this is the potential range of this species. Let me use my pointer. So Florida right here, as you can see, we are right on that border of where the snail may or may not be able to get to us. So that being said, with like changes in temperatures throughout the years, as time goes on with climate change, we might be able to see the snail move into populations such as Gainesville in the near future. Um, but as you can see, it is predominantly across the equatorial regions in the tropics, and where it is found is natively in Kenya and Tanzania, is their native range. So this is another map showing how the snail has moved across the globe. And so these bar charts are basically the diversity of the snails in that region of the world. And so if you look at Tanzania and yeah, well, Tanzania for now in the African continent, their diversity is high in this region. There are many different haplotypes, as we would call, of this snail. However, moving into, as the snail has moved across the globe, it's mainly only one snail or two snails that have actually caused it to become invasive and move. So the diversity across the globe of where it's found is not as high. For instance, in where's Florida? In Florida, there's only one haplotype that's ever been found in this part of the world, meaning that the snail is fairly not diverse and it's fairly inbred in that population. Um, okay, so with the introduction of new species into the environment comes new parasites and potentially new diseases. So we've seen this with things like COVID-19. We've seen this with leptospirosis, with rats moving across the globe. However, now we're seeing it in terms of with gastropods. The thing about gastropods is that they're really easily to go undetected, especially if you're transporting soil from one continent or one area to another. The eggs can be very, very small. So those eggs can go undetected in soil, and then once it reaches its destination, those eggs might hatch, and then they only need to have one egg in order to start a population because they can reproduce clonally. So they can be asexually reproducing. So when these snails are first born, they're all males. And then once they reach sexual maturity, they develop female reproductive organs, which then they can fertilize their own eggs and reproduce that way. So one egg only needs to go unseen for about 5 months to then start a population. However, with the spread of these snails comes new parasites. Because the snail moves across the globe, it picks up parasites as it stops from one country to the next. And interestingly, the parasite was first detected in Taiwan in 1944. However, it was never associated with a disease at this time. It was only associated with the disease about 2 decades later when they started realizing that this parasite was causing meningitis or a neuroangiostrongylosis, which is a condition where the parasite migrates to the host brain and causes an infection there, causing meningitis symptoms. Um, so the spread of the snail was most likely spread during World War II around that time, when there was increasing access to travel and transport of goods across the globe. This parasite is infectious to many species, so it's infectious to dogs, cats, apes, hedgehogs, orangutans, but their primary hosts are rodents, and their secondary hosts are gastropods. And I'll tell you a little bit about that moving forward as well. Okay, before I get into that, I'm just going to take a break and make sure, can everybody still keeping up with me as I'm moving forward? A gastropod is any, so it's a snail or a slug. It's any invertebrate that has a foot or only one body, of a muscular body that it moves with, basically. Um, so any gastropods can um, pick up this parasite. So this can be slugs, this can be any species of snails. Um, Pretty much that's it for these two. I wouldn't consider, yeah, that's pretty much it for these two. Okay, so Angiostrongylosis cantonensis is a primary host of rodents. So what this means is that the parasite is only sexually reproductive in rats, um, but it has to move outside of the rat before it can complete that life cycle. So the rodent, when the parasite lays eggs inside the rodent, it then defecates these eggs, and the eggs hatch in the soil, or they hatch, or they stay dormant until a gastropod, so a snail or a slug, comes and eats the eggs. Then the eggs hatch within the gastropod. This is when things get funky, because the gastropods, as they move about the environment, they crawl on produce, they crawl on surfaces that have, you know, they fall into water, and this can cause the parasite to be released from the snail and move on to produce or move on to into like water systems, and that's how people can then get affected with it. So there was one case in Jamaica where the snails were in a salad that some tourists were eating. And when they moved and went back to Canada, they all became infected with this meningitis symptoms. And it was tracked back to this one restaurant because they had very small neonate snails, baby snails, inside the salad. So that's one way that people can become infected. And this is known as a paratenic host. So it's a host that is not the actual or expected host of this parasite, but it still can survive in this host, and it still causes diseases in this host as well. So then the life cycle is completed when the rodent then eats this parasite, basically. And like I was talking about at the beginning of my presentation, I focused a lot on this One Health framework, which is connecting people with the environment and the science that we're doing. So I made it a really big part to understand if we had this parasite within snails in Barbados. Because even though the snails have been in Barbados for about 20 years, we had never actually tested successfully if they actually contained this parasite. And so that would be something that is very necessary for us to know and the people to support the health of people who live in Barbados. And so we use techniques such as PCR and gel electrophoresis, which I will not get into. But these bands tell us that this snail was infected with the parasite here, Angiostrongylisis. Okay, this is just how you write it up for a paper, and then it was published for open access so that anybody in the world can see that in Barbados we have this parasite, so that if travelers are coming or if science needs to be done in Barbados about this snail, they can know where to find it and who to contact about how it was determined that it was infected. Okay, I'm going to slow down a little bit. Okay, we're going to take a little back step and we're going to talk about something experiments that I did right here in Gainesville with a population of giant African land snails that are kept in quarantine at the Florida Department of Agricultural Services. So this is located, I'm not sure, does everybody know where this is located in Gainesville? So it's located, if you know where the Harn is, the Museum of the Arts Museum, it's right in front of that. So it's right connected to also what is the Natural Area Teaching Lab that UF has. So they kind of share that property in those boundaries. And so they have, when populations of giant African land snails come up in Florida, they collect a subsample of these snails from that population to run experiments in to test to see if they're infected with parasites. None of the ones in Florida are infected with the parasite so far, so that's good for us. But yeah, so this was how I housed these snails. And so this is just a little video of them. And as you would see, they are very large snails, so they can get about the size of your hand. They love water, and I kept them in these containers. Okay, so the first couple questions that I wanted to look into were about the impacts of these snails on economically important crops in Barbados. Um, so I looked at cucumber, okra, and sweet potato, as these are large, or these are things that are grown very readily in Barbados. And then I looked at the impacts of iron phosphate baits, which is a poison that is used a lot to kill the snails. Um, this is just a graphing of that experiment. So what we can take away from here is that they really like cucumbers, which are high in water. It may be this for a couple reasons. It may be that it's just not a huge source of nutrients, so they have to eat a lot of it. But they are able to eat a lot of it. And then the next one would be okras and then sweet potatoes. Surprisingly, these are not that different, but if we're looking at the cucumbers, you can say they tend to have a preference for certain types of foods. So this might be informing farmers like what is better to plant in their land or what needs more protection from these gastropods, the snails, than others don't. This was also the impact of the molluscicides. So you can see that it did decrease their amounts of food that they ate in the cucumber treatment over time as well. So the molluscicide treatments do work. However, the poisons that they use for snails are not specific to any one snail. So if you put a poison for a snail down expecting to only kill the giant African land snail, you'll kill all species of snails in your area. So that's not really the best when it comes to conservation and conserving certain species over not. Okay, the next thing, which I thought was really interesting, the question that I had at the FDAX was, is there a preference for settling location across a chemical soil gradient? And I'm thinking about this because of the differences in calcium levels in Barbados. So Barbados is formed by limestone deposits. As one tectonic plate moved under the other, calcium carbonate built up in that rift of the tectonic plates. And as magma and movement happened, it formed Barbados, and that's how Barbados came about. So we have a lot of calcium. We don't have a lot of soil per se, but right underneath that is a layer of calcium sedimentary block, which then leads to calcium being in our soils, which snails need to grow their shells. So that's why it's an important nutrient for the snails. Um, then it's surprising, Walter, that you brought up St. Vincent, because in 2020, La Soufriere volcano in St. Vincent erupted. And what happened, because we are due east of St. Vincent, the winds brought the ashfall from the Soufriere volcano into Barbados. And so we had buildup of sulfur, buildup of this sedimentary volcanic ash that was layered all over the island of Barbados. And I was thinking about it, and I was wondering, this has to impact the wildlife in Barbados in some way. It's dumping huge amounts of sulfur onto the beaches, onto the gardens, and this has to impact snails in some way. So I ran an experiment where I housed snails in different treatments of sulfur gradients increasing up, calcium gradients increasing up, and then a control, of course, to make sure that nothing was different. And I kept them in these containers over here for 3 weeks. So they were housed 1, 2, 3, 4, 5, and each started in a different treatment and then was rotated each day to determine, which treatment they tend to like. And so this is just a plotting of that. If you can visualize the container, so um, this container here with the 5 blocks basically is a representation of this. So the blue, or the bottom graph, is if the snail was not on the soil at all. If the snail was on the top of the wall or on the sides of the wall instead. And the calcium was if increasing levels of calcium and increasing levels of sulfur going down, if that makes sense. And then these grays are the controls, where it was just soil. There was no calcium or no sulfur added. And we can kind of see that they do have a preference that they tend to avoid both calcium and sulfur, and they would prefer to be off of that type of soil, because you can see that in the controls in those environments, they did still prefer to be on soils where there were no calcium or sulfur in the soil. However, um, you can also attribute that to being a sign of thigmotaxis. And thigmotaxis means basically when an animal prefers to be along the border of a container. So if you think about a cockroach or a roach in your house, you typically see them running across the walls, the sides of the walls, because that's a safe environment for them. They know that they can like hide in the crevice of the wall, and that's known as thigmotaxis. So that might also determine that snails are showing signs of thigmotaxis when in a risky environment as well. Because you can see that in the control environments, they are also settling. This 20% is the other side of the container. This 10% is the other side of the container, and this 9% is the other side of the container. And you would assume that because they're avoiding the sulfur here, they're like really low percentages in the in this sulfur, that they would also want to avoid the 10%. But maybe they're in high percentages in that because they're trying to get out of the sulfur and they're not on the walls as yet, if that makes sense. Yep. Okay. So moving off from this experiment now and moving back into Barbados work. And of course, those experiments before informed what I was going to be doing in Barbados moving forward. But I really wanted to ground my research in what the people in Barbados actually want and need. And so I determined that I would have to do some sort of qualitative research to determine this. So I did I constructed an interview that I would conduct with farmers and gardeners that went through a few questions with them. It was about a 15-minute interview maybe that I then was able to listen through and see what their issues are having. So some of the things that I looked at were um, if they what control methods they use. So what poisons they used, if they like move, if they killed the snails themselves, what they kind of things, what invasive animals they see on their properties, and general like associations with the gastropods along their properties. Um, so some preliminary findings basically showed that farmers, of course, who are actually using farming as their own livelihood, viewed the snails as a negative thing, of course. They used a lot of poisons. So they used metaldehyde baits, um, which are heavy metal poisons that go into the soils and basically stop the snails from eating. So it stops their, no, it paralyzes their not salivary glands, but it's their like secretory glands. So it stops them from cooling themselves down, and basically they paralyze and overheat. But like I said, this also kills lots of other animals, and this is also a bait that will also poison your dogs, poison other animals. So it's not great to be used, but it works really well for snails, and it's fairly cost effective in Barbados. So farmers will dredge the soil with it are some questions that they used. So they place metaldehyde poisons before and after they plant to make sure that no snail comes into the soil that they're planting or leaves. But if you can imagine, if they're putting this heavy metal poison into their plants, they're also killing many other microbes and other things in their soils and potentially causing problems for people who are eating the produce afterwards as well. However, if you look at people who just kind of have a home garden, they don't really rely on the land as their primary source of income. They typically see the snails as a reason to get outside, as a reason to attend to their garden, as a way to make sure that their plants aren't going to be eaten by the snails, basically. Um, they don't like them, but they have learned to live with them, basically, was the understanding that we came from. Okay, so then from that was the final part of my dissertation research now, which is looking at a non-chemical strategy to removing these snails from people's properties. So instead of us having to use all the poisons on the properties, we're going to try and use a non-chemical integrated pest management strategy, which is a long thing, but basically it is these cardboard made boxes or cone boxes, which I'll get into a little bit, which allow the snails to migrate inwards, and they have a mixture of flour and yeast that is fermenting sugars, which will basically attract the snails to those cones and provide them a nice place where they want to be so that then people can come and remove the snails from the environment and the specific snails that they don't want on their property, basically. Um, okay, so let's get into it. Okay, so the research questions, as we do in academia, are can a non-chemical aggregation device be used to attract invasive snails in agricultural, urban, and wild sites? The second one being, does color and placement of the device impact snail use? And so as you see here, there's one that's striped, red and white, and one that's only white. So there was some literature stating that snails can differentiate between high contrast colors. So maybe they would have an association with the red and white trap more so than the white trap because they can see it better. That was a potential hypothesis. And then the third research question is when are aggregation devices most likely to be utilized? Um, okay, so an overview of gastropods or snails in Barbados, and this is only a very, very small subset. But we have many species of native gastropods. This is our endemic snail. which means that it's only found in Barbados. The snail is not found anywhere else in the world, and that would be the Pleurodonte isabella or the Barbados blue snail. These other two snails are also native snails of Barbados that are not endemic but are found in other parts of the Eastern Caribbean as well. Whereas these snails are invasive to Barbados. So we have the giant African land snail, the Sagitina fullica, Bilimulus guadalupensis, which is a snail found and native to Guadeloupe, and then Zaccharizia provisiaria, which is a Cuban tree snail as well, invasive to Barbados. And in the interviews, farmers said that the Cuban tree snail actually was a lot more destructive than the giant African land snails, mainly because the Cuban tree snails are not only nocturnal, so they eat throughout the day as well. And they are smaller snails, so they tend to evaporate and desiccate slower than the giant African land snails will, because the giant African land snails have a lot larger surface to area volume, so they evaporate at a larger rate than the smaller snails would. The water within their body, basically, because snails are about like as humans are, but more so than they're about 90% of water, basically. Okay, so what is gastropod aggregation behavior? So gastropods aggregate for many reasons. They aggregate using each other's slime, so they can pick up on chemicals within the slime of other snails and follow those trails to find each other. What, such as like fish in schools or birds that flock together, it's a way to protect themselves. So it reduces predation risk. And also, like I said, because snails are 90% water, them grouping together actually buffers them from environmental stress. So if it's really hot in the environment, you might see a lot of snails grouped together to kind of buffer what one snail would be losing in like water mass. The other snails would not be losing, as if they were grouped together, they wouldn't be losing as much, sorry, as if they were by themselves, basically. Um, and this also, so as I said, also helps with temperature dependence. So if they're following in on those chemical cues, they can tell where healthy snails are going to hide out in certain risky environments and places, basically. So the second um, behavior is the homing behavior. And basically, this is the tendency of snails to return to a specific resting location. And so what this means is that snails have a home, and on quotes, what is seen as, so at night, they're nocturnal, they go out and forage, but in the daytime, they have a place that they'll come back to every day to stay during the day and rest, basically. Um, and so this is basically distincted by different foraging and resting patterns, basically, and they have a clear separation of activity and recovery areas. It might also be age dependent. So younger snails tend to move and find a home. As you would think, an 18-year-old child moving out of the house has a tendency to travel further than would an adult who has kids and is, kind of has their roots in their place and where they're going to live, basically. So it's kind of similar. It could be for snails as well. And also, this is a good use of resource acquisition. So if you have a home environment, you know what resources and what food is in that environment, and you know how to find it. Okay, so yeah, so this is one picture of a snail in its sandy home environment. And so this is a marked snail that I did for this experiment that I'm going to tell you about. And then this is another marked snail that's on the move at nighttime, basically. And so you can see that they do typically have, and this snail, I saw this spot multiple times over the week that this experiment was going on. Okay, so these are the traps. So as you can see, these four snails, so 1, 2, 3, 4, are in the tops of these cones here. So this is where they would crawl up And then when somebody came to check these traps, they would remove those snails, kill those specific snails, and leave any other snails that were actually, you can see a small little other species of snail right down here, but I cropped it out. And then within them are these containers with yeast and flour, which are fermenting sugars, which the snails feed on, such as, uh, rotting fruits and produce is what that simulates basically. And then, oh, we have the cat. And then we had temperature loggers that were inside and outside the traps as well, trying to see if there was a difference between the outside and inside traps. And that was basically one of the reasons why we thought the snails would use these traps is because the inside temperature is cooler than the outside temperature. Um, and so the traps were placed in groups of two, striped and non-striped. Colors were grouped for analysis. And then the four were placed under a tree and four were not placed under a tree for this shade of canopy versus non-shade of canopy, basically. Um, they were all baited with this flour, water, yeast mixture. And then comes for the very exhausting, tiring part was I checked these traps daily for 5 days each, at 8 a.m. to 9 a.m., at um, 8 p.m. to like 8 p.m. to 9 p.m., and then 2 a.m. to 3 a.m. basically. So every day for 3 weeks, I was waking up 3 times a night to basically track those traps, which was great fun, but I would definitely not do it again unless I had help. These were the three locations across the island where the traps were set up. So this was a agricultural site, this was an urban site, and this was a wild site across the island. So temperature readings for the habitats, you can see that the red is the outside temperature and the blue are the higher line, or the lower line is the inside temperature. So when the peaks of the day at noons were the highest, about 45 degrees, 35 degrees Celsius, the traps did stay cooler on average than the outside temperatures. However, at nighttime, the inside and outtime temperatures were similar. And so potentially, as a bit of a getting ahead of myself, um, the snails did not stay in the traps during the daytime hours because it was the same temperature inside the traps as it would have been outside of the traps. So it just doesn't make sense for them to stay in there. They did use them at night though. So they did come get the baits and then they moved back out basically during the day. Um, in the agricultural sites, snails tended to use the traps a lot more than urban and wild sites. This might be because the snails are um, agricultural pests. They are more adapted to finding food in agricultural areas and in areas where the food resources are changing regularly. So when they don't have food in one area because the crops were just tilled, then they can move to another area and find food as well. So they might be more adapted to just finding resources, and they maybe found the traps in the agricultural sites better than the snails in the urban and wild sites potentially. Um, this is just an example of the amount of times that the snails were captured. So you can see that typically snails were captured only once, 1 to 3 times. But we did have snails, and this was the same exact snail because I marked them up to 7 times within that week, basically. So this does also show that snails have that like homing ability to go back and forth to the same environment if they find a food resource that they really like, they'll keep going back to it basically, which shows that if you keep these traps in your environment, they might actually be able to like over time help you reduce the population of snails if you're checking them regularly basically. Okay, this is another example of the morning, evening, and night, basically. This is just a plotting of that, of the differences of number of snails that use the traps across these time points. And basically, this is getting at if the traps are more effective or when you should basically check the traps um, more frequently versus not. And this basically shows, this is a walled z-test. I'm not going to get into a lot of the stats. It's a negative binomial generalized linear model, basically saying that you should check your traps at night, or at least in the evenings into early mornings, because the night traps, sorry, the morning traps into evenings when it's hot in the day, there are not a lot of snails in them. That's basically what this shows, because in the morning, as you can see, there are less snails, so only 2.5 on average versus about 8 snails on average for the night and evening time points. So unfortunately or fortunately, farmers should be checking these traps if they were to use them really early in the morning. But farmers, for the most part, are going out onto their yards really early in the morning anyways, because they don't want to be in their properties when the sun's hot. A lot of harvesting happens when the sun is now starting. So this is actually pretty good, and it might fit into their schedules to use on their properties more so than more than not. Okay. Remember when I talked about the difference in canopy, so placing the traps under trees versus not under trees? That didn't have a difference in if snails use the traps or not. Moreover, if the treatment was striped or not striped, also didn't have a difference on whether a snail used the trap or not. So that's good. That means that we can make it more of a streamlined trap that is fairly easy and cost effective to make. Um, Okay, this is just how I did my capture analysis. This just shows that there was a difference in location type of when the trap was to be used. This basically just shows that the traps were more effective on agricultural landscapes versus urban or wild landscapes. So it was the traps were better at aggregating snails on agricultural landscapes. Okay, more visualizations. Not going to get into that. Also, we're running down. Okay, cool. And then this is synopsis. So, can a chemical non-aggregation device be used to attract invasive snails in agricultural, urban, and wild sites? Maybe. So further testing is required to fully answer that question. However, aggregation devices are potentially more effective in agricultural settings versus urban and wild settings. Does color and placement of the device impact snail use? No, color and placement don't impact snail use. And then when are aggregation devices most likely to be utilized? Traps are most effective, like I said, during the evening and night time points. Okay, cool. And with that, I want to give a big acknowledgement to my lab and a really huge acknowledgement to Jane Brockman, who is a part of this community here and who invited me to speak. I received 2 grants that are in her name at the biology department for $3,500 totaling, which was allowed me to do most of this research. So Jane is the reason why I was able to complete this research and go back home as well with some sort of money and with the information and the collaborative network to complete this research as well. And with that, I'll leave you with a video of, so you were asking about what tells you a gastropod. So this is a gastropod foot moving. So this is the underside of a snail and what looks like a conveyor belt. So that's kind of how their muscles move as they're moving across a surface, basically. Yeah, and with that, I think I'm a little early, but I think that's it. Yes.
Thank you. You said that they forage for food at night,
Mhm.
and yet that's the time when they most use these cage kinds of things.
Yes.
That seems a contradiction.
No, so they are foraging. The traps themselves are foraging. It's food because the bait that we put in the traps is the flour and yeast, which is a fermenting sugar. Then so they're moving away from the crops to eat this instead. Yeah,
I apologize, I wasn't here for the very beginning.
That's
Did
okay.
you mention the density of the snails in the different environments?
I did not, but all the sites that I used are had similar densities. So I chose those 3 sites for the traps at to have similar densities across the sites.
I'm trying to figure out why you would get so many more on an ag site if you have the same density as the urban. It doesn't seem,
So the only
it's not intuitive.
it's not intuitive, right? The only thing that I could think of was that, so as to answer Mary's question, this flour and yeast debate is a food source. In agricultural environments, because the crops are moving and changing so frequently, I think the snails there are more adapted to finding food, or when there's a strong resource of food in their environment, they take that chance and they go and get that food while it's available versus in an urban and wild site, the habitats don't change as frequently. So where they would find food in an urban and wild site is predominantly the same throughout the year. So maybe that's why they don't have to, if there's a resource of food, they don't have to go to it in those sites. That's what I'm thinking. Yeah.
Other questions? I have one. I'll be last.
Okay, so
Okay.
she asked a question. I was going to ask about the concentration of the animals, but Mike, if they're not coming to the traps
Yeah.
in the urban, what are they eating? Do you
Well-
know what they are, going after? Is it garbage? Is it, what are they doing there?
Yeah, so I think it depends on what is in that environment. So in some, I can talk about the different sites because in the sites that I checked that were urban environments, if somebody had a mango tree in their yard and the mangoes dropped fruit certain time of the year, the snails were in that area a lot more. Yes, if there was garbage and there was a lot of fruit on the ground, the snails tended to be in that area as well. I also think that there might be a connection between where rodents are and where snails are as well, especially if they're in an environment where it's scarce of food, because they'll also eat the feces of other animals. So they might queue into where rats, where dogs defecate as well, and then eat those feces as well. That's in environments where there's not a lot of other food like rotting fruit and that sort of stuff that they might be doing that. So that's maybe what would change them.
Are there any good snails?
I think
I-
all snails are good snails.
No.
They
I
just
mean,
get a bad rep.
Do they have any redeeming value in agriculture, except for escargot?
In agriculture to be used, there's not real many uses for snails to benefit agriculture. There are uses for snails in other sectors, in terms of like the beauty industry. People will use the mucus from snails as like a facial treatment. People think that it will, help you increase collagen in your skin, and make it tighter or whatever over the years. But for actual agricultural uses and apart from escargot, there's not much benefit per se, other than providing a food source for other animals as well. And they aerate the soil. So when the eggs are in the actual soil and then the snails come up, it will disturb the soil, which will cause it to be aerated and allow other animals to get through. So maybe that's a benefit.
Okay, here's my question. Before we started, you said that the snails originated in Zambia or Kenya,
Tanzania and Kenya.
and that they eat the snails as their escargot. How about in the islands or other places around the world? Are they edible or eaten?
So when they first came into the Caribbean, they were brought into Martinique, and Martinique is a French territory. So they were brought in for food initially to the Caribbean islands, and that's then how it moved across the Caribbean world. We don't typically eat them in French territories. They do eat them in places like St. Vincent. They will eat them in places where there's a more predominant culture to eat what is called bushmeat. In Barbados, though, because we're very urbanized, we don't really eat a lot of like wild meat. We tend to only eat farmed meat, so we don't eat it in Barbados.
Thank you, and
Yeah.
thank you very much for coming. But you have some show and tell
Yeah,
things,
I
so
guess we can
if people
pass
want to
it.
come up and have a look, that would be great.
Yeah. And then,
One more question.
yes. I also have this fact sheet of like what to do if you see a snail in Florida when you're here and who to call. So if you want to take a picture for the numbers and stuff, you can also take that. Yes.
Okay, one more question. If the goal is to get rid of the snails, you're able to catch them in the agricultural environment.
Yeah.
Well, that's good. But if there's still the same concentration in the other environments and you're not having any luck, any ideas about how to capture them in the other areas?
Vigilance.
No, not yet.
Vigilance. You just have to be
Yeah.
very vigilant, really. Yeah, exactly. That's really it. The problem with the snails in Barbados, and Florida hasn't reached this level yet, is that they've reached a point in Barbados where we're never going to get rid of them. They're going to be in Barbados regardless. But if we can reduce the ways that they can cause infectious diseases, so if we can remove them from agricultural areas, we can probably reduce the amount of times that people are getting infected from these parasites potentially. In the urban environments, people tend to be very vigilant, so they tend to remove them from their properties more if they need to. But some people are kind of like, they don't mind them being there, they don't cause a big issue. So it depends on your outlook of things for yeah, basically. But, yeah. And so I can pass this around or like show this. These are what the snails actually look like. They're really large.